Hello, my name is Wissal Medjaouri. I'm in 6th grade and I'm from Al Huda School. My project title is Microbes and Sunbeams Finding the Ultimate SPF Shield. Ultraviolet rays can be very harmful to living organisms. Humans use sunscreen to minimize the damage ultraviolet rays can cause. While there are many sunscreen products used to protect against the damaging rays of the sun, choosing a higher sun protection factor is not always best. The purpose of my project is, does a higher sun protection factor mean better protection? The objective of this project is to check if a higher sun protection factor is better for human skin by testing it on a non-pathogenic strain of E. coli bacteria used as a bioassay. The origin of my project is that my mom has very fair skin and she never leaves the house without sunscreen. She struggles which SPF to buy, and I thought it would be a good idea for a science fair project to check which SPF works best. My independent variables are the degree of sun protection factor SPF 15, 30, 50, and 100. My dependent variables is the number of E. coli bacterial colonies cultured. My controlled variables are all cultures will be under the same temperature, which is 80 degrees Fahrenheit. All cultures will be the same distance from the UVA light source, and all cultures will be observed at the same time. I hypothesize that non-pathogenic E. coli bacteria is used as a bioassay under 395 to 405 nanometer light in petri dishes covered with different sunscreen SPF. Then the SPF 30 will show the most protection from the UVA light by allowing the most bacterial colonies to grow. The materials I used was the bacteria, which was a non-pathogenic strain of E. coli ordered from Home Science Tools website. The growth media was 15 pre-poured nutrient agar plates. The sunscreen samples were the same brands of sunscreen with different SPF values, which were SPF 15, 30, 50, and 100. UVA light source was 20 watts UV light fixture with clamp wavelength 395 to 405 nanometer. Incubation was a temperature-controlled atmosphere set to around 26.7 degrees Celsius or 80 degrees Fahrenheit. Is ideal for bacterial growth. The supplies were the sterile cotton swabs or pipettes, the plastic wrap or clear UV transparent membrane, a ruler to measure the diameter of the bacterial colonies, a permanent marker for labeling, and masking tape. The safety equipment was the nitrile gloves, the safety goggles, a lab coat or apron, and UVA protection glasses. To prep the bacteria, I removed the cap from the tube of broth included with the lyophilized culture. Then I removed the cap from the vial containing the lyophilized bacteria E. coli. Then, using the sterile pipette, I added about 1 milliliter of the broth to the vial containing the pellet. After that, I allowed the broth to sit for about 30 seconds to rehydrate the pellet. Once the pellet became rehydrated, I used the pipette to withdraw the broth and expel it back into the vial several times in order to break up the pellet and thoroughly mix the lyophilized organisms and broth. While using the pipette, I transferred the broth organism mixture in the vial back to the broth tube and loosely placed the cap back onto the broth tube. Then I incubated the broth tube containing the rehydrated organisms at the recommended temperature overnight. After the incubation period, I transferred the bacteria to the agar dishes using a pipette and a sterile swab. In order to conduct my experiment, I ordered a non-pathogenic strain of E. coli bacteria kit and 15 agar dishes. The first thing I did was I rehydrated the bacteria. Then I inoculated the bacteria onto each agar dishes. After that, I added the different sunscreen SPFs onto each trial. Then I added the petri dishes under the UVA light for 2 hours a day for 8 days. Every 2 days, I recorded how many bacterial colonies grew. This is my collected data. The E. coli bacteria cultured without sunscreen grew 6.33 average bacterial colonies and 6.67 for the average diameter. The E. coli bacteria with sunscreen SPF 15 grew 71 average bacterial colonies and 2.33 for the average diameter. The sunscreen with sun protection factor 30 produced the most bacterial colonies, which means that it had the most protection from the UVA light, with 116.67 average bacterial colonies and 2.67 being the average diameter. The sunscreen with sun protection factor 50 grew 78.33 average bacterial colonies and 3.33 for the average diameter. E. coli bacteria with sunscreen SPF 100 grew 71.67 average bacterial colonies and the average diameter was 3. If you look at the graph, you can see that SPF 30 produced the most bacterial colonies, which means that it showed the most protection from the UVA light with 116.67 average bacterial colonies, and the control, which had no sunscreen on the petri dishes, showed the least protection from the UVA light because it produced the least average bacterial colonies with 6.33 average bacterial colonies. SPF 50 came in second, SPF 100 came in third, SPF 15 came in fourth. In conclusion, the experiment supported the hypothesis. The Escherichia coli bacterial growth for the sunscreen with sun protection factor 30 produced the most bacterial colonies after 8 days of subjection to UVA light for 2 hours a day. The sunscreen with the sun protection factor 50 came in second, SPF 100 came in third, and SPF 15 was fourth. The least bacterial growth was the control, which had no sunscreen on the petri dishes. The use of Escherichia coli as a bioassay was to determine what sunscreen SPF best protects against UVA light proved that a higher SPF is not necessarily better. Sun protection factor 30 worked the best. Thanks. I would like to thank my mom for buying me the kits for my project and supporting it. I would also like to thank my teacher for supervising me while doing my project and helping me for the things that I did not know how to do.